Good morning, everyone. My name is Francisca Suárez and I work in the area of microbiology of the University of Almería. The title of my presentation is Suppressive Capacity of Compost Against Plant Pathogens, Search for New Biological Control Agents. Before starting, I would like to thank Dr. Miguel de Cara for the opportunity of being here at this interesting webinar. I'm part of the research group Bio175 named Development of Microbiological Processes to Improve Soils of Agricultural Interest. For 20 years, most of my research work has been carried out in relation to the microbiota of the composting process. Our group has a wide experience in all aspects related to composting of agricultural waste. Specifically, our working topics are valorization of horticultural wastes, composting microbiota, and lignocellulolitic microorganisms, antagonism and suppressivity towards phytopathogens, and search for microorganisms producing substances of agricultural interest. I want to highlight that the search for biological control agents from natural substrates, but especially from compost, is one of the research areas in which our group has heavily worked. The content of this session includes general aspects concerning the biological control of plant diseases, mainly focused on typical horticultural crops in the southeast of Spain. Besides, I will show a quick review related to microbial action mechanisms directed to plant growth promotion and plant pathogen control. Finally, I would like to tell you about the experience of the research group Bio175 at the University of Almería in relation with the search for new biological control agents from natural substrates, but especially from compost. Currently, environmental security and safety for consumers of vegetables and fruits are both very important aspects which concern the companies in the agri-food sector, since both of them will be directly related to the quality of their products. In this sense, the most common problems in agriculture are unfavorable weather, weed growth, entry of insect pests, and the emergence of diseases caused by microbial phytopathogen agents. All these aspects cause economic losses for farmers, prices are increased in the market, and the global availability of food is reduced. In relation with the last point indicated, plant diseases need to be controlled to maintain the quality of food. Many phytopathogen agents affect several crops around the world. Crops of solanaceous and cucurbitaceous, as well as citrus, cereals, or tropical crops, are damaged by different phytopathogen bacteria and fungi. This slide shows some of the most important diseases caused by bacteria and fungi in the southeast of Spain. We can see here bacterial wilts and spots, anthracnosis, damping off, or different fruit rot caused by several aggressive fungal agents. Different strategies may be used to prevent, mitigate, or control plant diseases. The most traditional solutions are crop rotation or the use of resistant seeds, chemical fertilizers and pesticides, or the use of suppressive substrates such as compost. Despite good agronomic and horticultural practices, growers often rely heavily on chemical fertilizers and pesticides. Don't forget that such inputs to agriculture have contributed significantly to the spectacular improvements in crop productivity over the last 50 years. For instance, this is the particular case of the province of Almería and its famous sea of plastic. However, the environmental pollution caused by the excessive use of agrochemicals has led to considerable changes in people's attitudes towards the use of pesticides in agriculture. Today, we all know that the use of agrochemicals for decades has led to negative effects on health soils, and environment. Consequently, some pest management researchers have focused their efforts on developing other alternatives to synthetic chemicals for controlling pests and diseases. Among these alternatives are those referred to as biological control. Biological control using beneficial microorganisms has been considered a viable alternative for the replacement of chemical methods. The most common antagonist agents used to control plant diseases include bacteria such as Pseudomonas, Bacillus, and Burkholderia, fungi such as Trichoderma, or even microalgae and cyanobacteria, whose presence in soils and compost are very significant. They are involved both in the suppression of plant diseases and plant growth promotion. In fact, many studies have been undertaken in order to analyze in depth possible interactions among compost and rhizosphere and plant pathogens. Therefore, it will be very important to know and understand the mechanisms by which all of these microbial species interact with other plant pathogens. We can see in this slide some of the most recognized microorganisms involved in the biological control of phytopathogens. These include fungal genera such as Aspergillus, Gliocladium, Pethylomyces, Penicillium or Trichoderma, or bacterial agents such as Phantoea, Pseudomonas or Bacillus, or the genera Streptomyces, which is considered an excellent candidate as biological control agent. In general terms, all these microorganisms could be called PGPM, derived of the concept of plant growth promoter agents. They are involved in various mechanisms to promote plant growth and control plant pathogens. In this slide, you can see the different microbial mechanisms to improve plant growth. I would like to stand out the microbial mechanisms related to iron solubilization, potassium and phosphorus availability, nitrogen supply, or the production of phytohormones, vitamins, and amino acids. In this sense, the presence in the compost of all of these microbial groups support the beneficial effects on this humic amendment after its application to the soil. The reduction of the incidence of many plant diseases can be explained by one or more biological effects, which are summarized in the application of suppressive compost by foliar or soil application. This scheme presumes the general mechanisms of control of plant pathogens. In general terms, the biological control can be exerted directly on the phytopathogenic agent itself or through the induction of plant resistance mechanisms. On the other hand, the application of post-suppressive substrates and microbial inoculants could also act as an important stimulus for plant growth. Based on everything indicated above in relation to the application of suppressive compost or PGPMs, I would like to tell you my personal experience in relation to searching for new biological control agents from natural substrates, in particular from compost. Composting is the biological decomposition of organic matter under controlled conditions into a humus-like and stable product called compost. The microorganisms naturally occurring in the raw materials are the main responsible of this biotransformation. Among the benefits of the application of compost to agricultural soils highlights the suppression of telluric and foliar plant pathogens, as well as the promotion of the plant growth Then the biological properties of the compost remain responsible for both effects. For more than 15 years, our group has characterized a valuable collection of strains, bacteria, fungi, and actinomycetes in relation to their capacity as biopesticides, phytostimulants, and plant resistance inducers. We have tested near of 2,000 microbial isolated towards the in vitro growth of bacterial and fungal phytopathogens, such as Fusarium oxysporum melonis, Pythium ultimum, Botrytis cinerea, or Clavibacter michiganensis. The technique traditionally used to detect biological control agents is called dual culture. However, in this case, it must be taken into account that the rest of the components, plant and soil, don't participate in this interaction. All of these strains have been also tested as producers of agricultural substances of interest such as salicylic acid, siderophores, cyanide, chitinase activity, and phosphate or potassium solubilization in vitro. Several isolates have been too investigated in relation to their capacity to produce phytohormones such as indolacetic acid, zeatin, abscisic acid, jasmonate, or salicylic acid. Besides production of vitamins and amino acids such as niacin, thiamine, biotin, vitamin B12, pantothenic acid, and cysteine was also investigated. Much of these microorganisms from suppressive compost have been also tested in relation to their capacity to stimulate the seed germination or control foliar and vascular pathogens in vivo. Definitely, all these tests have been useful to corroborate the great potential of the compost microbiota to suppress plant pathogens and promote plant growth thanks to various mechanisms that directly affect the plant or the pathogen. Finally, I want to show here a photo of my co-workers. This research group is made up of 9 persons, including professors, postdoctoral researchers, and predoctoral students. Currently, the director of the group is Maria José López, but I would like to make a special mention of Professor Joaquín Moreno, recently deceased, who has led the Bio 175 group for 30 years. He was an excellent microbiologist and scientist and a great expert in composting process. Thank you for your attention, and I hope to resolve your doubts in the round of debate.